Assalamu alaikum my dear students, welcome to STUDYHIGH.Pk and subscribe keeping contact for more videos and lectures. Today I am going to start discussion of lecture 3 of chapter Lipids, Medical Biochemistry. Two very important topics that I will discuss with you in this lecture are eicosanoids and its types and prostaglandins, Nicotrienes, lipoxins, their synthesis, formation, role in causing different type of diseases in the human body and their cure as well as. So first of all, I want to inform you the learning outcomes of this lecture. What are the important concepts that you will came to know after completion of my discussion on this lecture? Like C1, clinical number 1, how eicosanoids differ from other hormones functioning. Eicosanoids are produced by almost every cell of the human body. Almost. How their function is different from other type of hormones because eicosanoids are also hormone like enzymes. They act like hormones. So what is the difference between other type of hormones like insulin, glucagon and those of eicosanoids? C2. How arachidonic acid synthesized in our body. You must know arachidonic acid is a derivative or precursor of all types of eicosanoids that are produced in our body. So you must know from where this arachidonic acid entered or comes in our body or what are the important sources of arachidonic acid. C3. What is the role of phospholipase A2 and linoleic acid in eicosanoid synthesis? C4. How eicosanoids cause inflammation, asthma and their role of different type of eicosanoids in uterus, in childbirth, in menstruation, lungs, kidney, etc. C5. Role of aspirin and cortisol in treatment of the patient that are suffering from asthma, inflammation, etc. Difference in function of thromboxane and prostacyclins. These are some important concepts that you have to know or you must remember after completion of my discussion on this topic. So let's start discussion on our lecture 3 of medical biochemistry. First of all, I want to inform you what is eicosanoids. Eicosanoids consist of prostaglandins, Nicotrienes, lipoxins. And we define eicosanoids as the compounds that are acting like a hormone or cell signaling molecules. Cell signaling molecules, prostaglandins, Nicotrienes and lipoxins that are derived from this arachidonic acid. What is arachidonic acid? I have explained you in detail in the previous lecture. A polyunsaturated fatty acid that consists of 4 double bonds at position 4, 8, 11 and 14. So this is arachidonic acid that belongs to omega-6 family. All these eicosanoids are derived from this arachidonic acid, whereas prostaglandins further consist of 3 classes: prostaglandins, thromboxanes and prostacyclins. Why we call prostaglandins or prostaglandins? I want to inform you the history of prostaglandins. The first time prostaglandins are isolated or extracted from the fluid produced by the seminal vesicle, produced by the prostate gland. Prostate gland produces a fluid from which the first time prostaglandins or prostaglandins are extracted. That's why, because they are produced by the every cell of our human body, but because they are first identified in the seminal vesicles or the fluid produced by prostate gland, that's why we call or name them prostaglandins by the scientists that discovered them at that time. And how the function of these hormones is it differ from other type of hormones because these are also cell signaling molecules. The function of these is different from other type of hormone in that way. Eicosanoids are produced by almost every cell of the body. Almost every cell of the body in small amount. whereas the other type of hormones like insulin as well as glucagon has specific site of production, like insulin is produced from the pancreas, whereas the other type of hormones like those that are derived from hypothalamus or thalamus as well as those that originate from pituitary gland. Every hormone that is functioning in our human body has a specific site from which it produces, and it also has a specific site at which it acts, like insulin produced from pancreas but it works in liver, whereas in the same way the follicle stimulating hormone secreted from pituitary gland but it acts on the uterus. This is the difference between eicosanoids and other type of hormones. Eicosanoids are produced from every cell. They have not a specific site of production or specific site of action or protecting. They are not produced by the glands. So this is the difference. A second difference is that eicosanoids have short lifetime. They destroy soon after their formation, after their working. Whereas these hormones, some have very long time functioning in the body, some destroy after some time after completing their functions. So these hormones has different role, or they are different in number of aspects from the eicosanoids. Now I am going to discuss the second important concept that I have tell you will be know after completion of my discussion on this topic: how arachidonic acid synthesized in our body cells, and what are the sources that involve in providing arachidonic acid to our body cells so that our body cells can produce prostaglandins, licotrienes, lipoxins from the arachidonic acid. First of all, you must know that our body consists of thousands of cells. Every cell consists of a cell membrane. I am showing the cell membrane of this cell here. Every cell consists of a cell membrane that consists of phospholipid bilayer. Different type of phospholipids are present in our body cells. Some phospholipids are phosphatidylcholine. Some phospholipids that are present or related to our cell membrane consist of cholesterol. So our body cell membrane consists of different type of phospholipids. Some may contain choline as a base that attach with a phosphate group of phospholipid. A specific type of phospholipid is also present in our body cell membrane, and that is called phosphatidylinositol. Phosphatidylinositol also consists of glycerol, and two fatty acids attach with this glycerol carbons at carbon number 1 and carbon number 2. Whereas at the carbon number 3 of this phosphatidylinositol, there is a phosphate group that attach with the inositol ring. So this is the structure of phosphatidylinositol. Whereas at carbon number 1, the acid that attached with the glycerol of phosphatidylinositol at carbon number 1 is stearic acid. Whereas the most important acid that attached with the carbon number 2 is arachidonic acid. So here arachidonic acid attached with the carbon 2 of phosphatidylinositol, and the detailed structure of phosphatidylinositol we will discuss in our coming lecture. So what will happen? This cell membrane also contains a number of receptors. When some hormone bind with this receptor, it will also result in production or cause or release of a hormone enzyme phospholipase A2. We will discuss in detail what the process of phospholipase A2, A1 in different type of reaction. So when a hormone bind with this receptor, it causes the release of phospholipase A2, and then phospholipase A2 causes the release of this phosphatidylinositol into the cell or cell membrane, and the further process occurs is that this phosphatidylinositol is broken down into two components. One consists of glycerol diacyl glycerol. This is one component that is broken, and second is phosphate and inositol. Then it is phosphorylated and converted into inositol triphosphate. So these are the two components that then further be entered into our cell and act like second messenger system, act like other type of messenger that carry information and cause a different type of increase in intracellular calcium and different type of functions that we will discuss in coming lectures. But here is arachidonic acid. Phospholipase 2 cause the release of arachidonic acid from this phosphatidylinositol, and this arachidonic acid can further be used by the cell for the production of these eicosanoids. So if someone asks you, or you may think, what are the sources of arachidonic acid? The first is the phosphatidylinositol. And what is the role of phospholipase A2? This is phospholipase A2. Role of phospholipase A2 is that it involves in release of arachidonic acid from phosphatidylinositol, that is also a component of phospholipids of cell membrane. In this way, arachidonic acid has been removed from the phosphatidylinositol, and this arachidonic acid that is connected with carbon 2 of glycerol of phosphatidylinositol can further be used for the production of different type of eicosanoids. Now I am going to inform you about the second source of production of arachidonic acid. You eat oil or peanuts, coconut, different types that contain a acid called linoleic acid. Here is a oil or some fruit that you engulf, eat. As a result, you obtain what? You eat or drink a diet that contain linoleic acid. The detailed structure and formula of linoleic acid I have explained you in previous lecture. It consists of all cis 912 octa-decadionoic acid. It consists of total 18 carbon atoms and 2 cis double bonds at position carbon 9 and carbon 12, and it belongs to omega 6 family. Oh, you eat this will be absorbed by our intestine. Then there are two processes that involve in conversion of this linoleic acid into arachidonic acid. It consists of 2 double bond, whereas arachidonic acid consists of 4 double bond. So first denaturation of linoleic acid occurs. As a result, there are 4 double bonds. We made linoleic acid into 4 double bonds. Unsaturation or denaturation desaturating enzymes are used for conversion of 2 double bonds of linoleic acid into 4 double bond. Then another process that happens is the addition of 2 more carbon atoms. Because arachidonic acid is icosa, it consists of 20 carbon atoms, whereas linoleic acid consists of 18 carbon atoms. That's why we further add 2 carbons into it. As a result, it will be converted, or we can then ourselves can convert you and use this as a arachidonic acid. So linoleic acid or the diet that contains linoleic acid can also be used for the production of arachidonic acid. Now arachidonic acid has been produced. You know what are the sources of arachidonic acid. Now I am going to explain how this arachidonic acid can be converted into different types of eicosanoids like prostaglandins, Nicotrienes, lipoxins. This arachidonic acid can be processed in two ways: through cyclooxygenase pathway or lipoxygenase pathway. There are two pathways in which arachidonic acid can be processed. One is cyclooxygenase and second is lipoxygenase pathway. In cyclooxygenase pathway, first this arachidonic acid that is present in U form is converted, or a liquid cyclopentane molecule has been created in this arachidonic acid. That's why we call cyclooxygenase pathway. And in this pathway, two type of enzymes: cyclooxygenase 1 that are involved in production or conversion of arachidonic acid into other types in some type of cells, and cyclooxygenase 2. There are two type of isozymes that involve in conversion of arachidonic acid into a new product form that will helpful in producing a number of prostaglandins. Cyclooxygenase enzymes, cyclooxygenase 1 or 2, these are two different type of enzymes, They will convert arachidonic acid into PG. PG means prostaglandin G2. This is also a type of prostaglandin G2. 2 means that it contains two double bonds in the side ring. So if this is a side ring of this prostaglandin, it will contain only two double bonds. Whereas in normal conditions, arachidonic acid consists of four double bonds. PGG2 is formed because of the cyclooxygenase enzymes. Then this PGG2 through an other enzyme peroxidase converted into PGH2. Once this PGH2 produced, all other types of prostaglandins PGE2, PGI2, PGI2 mean prostacyclin, thromboxans, all other types can be produced from this PGH2. So first, cyclooxygenase enzymes convert arachidonic acid into PGG2. Then peroxidase enzyme convert PGG2 into PGH2. Then from this prostaglandin H2, we can get any prostaglandin that you want. Once PGH2 produced by the action of peroxidase enzyme, this can be converted into PGI2, which is also called prostacyclin. By using an enzyme prostacyclin synthase, PGH2 can also be converted into PGE2 by an enzyme isomerase. This PGE2 contains carbonyl oxygen double bond at carbon number 9. This can be converted into PgF2 by reduction of this carbonyl oxygen into hydroxyl. So the only difference between PgE2 and PgF2 is that in PgE2 there is a double bonded oxygen at carbon number 9, whereas in PgF2 there is a hydroxyl group at this position. Thromboxans A2, thromboxans B2 can also be obtained from this PGH2, and PGD2 is also a sleep promoting substance, prostaglandin that can induce sleep in the patient. I wanna like to show you the detailed structures how this process is occurring. This is a structure of arachidonic acid, consists of 4 double bonds at 5, 11, 8, 11 and 14. Through the action of cyclooxygenase enzyme, it is converted into PGG2. In PGG2, there is a cycle formation, as you can see, cyclopentane, and there are addition of two oxygen molecules in this compound, and this bond has been broken and converted into carbon number 13. As a result, OO hydroperoxy bond formed at this carbon. This is an indication of PGG2. In the next step, peroxidase enzyme causes the conversion of this PGG2 into PGH2, in which there is a hydroxyl group. This hydroperoxy group has been replaced by hydroxyl group. This is a PGH2 formation. Once the PGH2 has been produced, it can be used for the production of anything you want. Like through the action of prostacyclin synthase enzyme, it can also produce PGI2, prostacyclin, who you can determine whether it is a prostacyclin or an other type of prostaglandin. There is a specific characteristic of prostacyclin. It consists of two cyclopentane rings, whereas all other prostaglandins consist of one. And second, prostacyclin or cyclopentane ring that present in prostacyclin also interrupted with oxygen atom. So this is the character of prostacyclin. Through the action of isomerase enzyme, PGH2 can be converted into PGE2, in which there is a double bond at carbon number 9 of PGE2. This is a very important. You must know the structure of PGE2 because it is the most abundant prostaglandin. It is a prostaglandin that is mostly in most abundant form, is also secreted by our kidney cells and involved in controlling the renal function or renal tube functions by causing vasodilation and different type of functions. So PGE2, E2, 2 means if 2 means there are two double bonds in the side chain. Here 1 and 2. Likewise PgH2, there are 2 double bonds in the side chain, 1, 2. This is the structure of PgE2 that consists of 2 hydroxyl groups connected at carbon number 11 and carbon number 15. Through the reductase enzyme, PgE2 can be converted into PgF2. This is the structure of PgF2, in which the only difference between the PgE2 and the PgF2 is that this double bond O has been reduced by reductase enzyme into hydroxyl. So this is also a very important F2 that involved in causing or producing, inducing labor induction in the uterus and uterine contractions. So PgF2, the only difference between PgE2 and PgF2 is the hydroxyl and carbonyl oxygen. PgD2, which is a sleep promoter prostaglandin, here is structure of prostaglandin PgD2. Isomerase enzyme causes its conversion. Thromboxanes A2 can also be produced from the PgH2. The structure of thromboxane A2 is that it consists its cyclopentane, not cyclopentane but cyclohexane. Ring is interrupted with oxygen, and oxygen is also bonded or present inside of the ring, as you can see. So this is the whole mechanism how arachidonic acid is used in production of different type of prostaglandins. Something that you must remember is the structure of PgE2 and PgF2, as these are very important in kidneys, uterus, stomach, etc. Whereas second thing, what are the enzymes that involved in conversion? As prostaglandins also cause inflammation in asthma patient and different type of diseases, then you have to stop the production of these prostaglandins that involve in causing inflammation. Then you have to stop any one of these enzymes, whether cyclooxygenase 1 or 2 enzyme should be blocked, or peroxidase enzyme should be blocked, or whether the production of arachidonic acid should be blocked, so that the inflammation should be reduced and the patient suffering from asthma or any type of difficulty in breathing should be controlled. Before discussing the working of aspirin, ibuprofen, NSAIDs, non-steroidal anti-inflammatory drugs, and cortisol, which is a steroidal anti-inflammatory drug, first I want to inform you lipoxygenase pathway, how it works in our human body. Like processing arachidonic acid through cyclooxygenase pathway, lipoxygenase pathway consists of 2 types of enzymes. And lipoxygenase pathway include the formation of hydroperoxy group or inducing the hydroperoxy group on the arachidonic acid. There are 2 types of enzymes that involve in lipoxygenase pathway or production of nicotrienes and lipoxygens. 5-lipoxygenase enzyme causes the conversion of arachidonic acid into 5-hydroperoxy icosa tetraenoic acid. Icosa 20 5-hydroperoxy. At 5th position of this arachidonic acid, there will be a OOH bond. So this 5-hydroperoxy icosa tetraenoic acid is also written in simple form in 5-heat. This 5-heat hydroperoxy icosa tetraenoic acid can further be converted into licotriene A4. Licotriene A4 by conversion of this 5-hydroperoxy group into removal of this hydroxyl from this. Removal of hydroxyl from the hydroperoxy group results in conversion of this 5-heat into licotriene A4. Like PG prostaglandin H2, this licotriene A4 can further be converted into any form you want. And other type of licotriene A4, B4, C4, D4, E4. Naming of licotriene is very easy. A, B, C, D and E. You can easily remember this. But the production is also very simple. Like 5-heat, in 5-heat the hydroperoxy group has been converted into O form. As a result, we obtain licotriene A4. Licotriene A4 can be converted into licotriene B4 by simple converging of this O into hydroxyl. When at the carbon number 5 of Licotriene A4 is hydroxylated, then we will obtain Licotriene B4. But when Licotriene A4, when we add a 3 amino acid complex, that after addition into Licotriene A4 will give rise to Licotriene C4. The complex that we add is glutathione. This is a 3 amino acid complex. It consists of glutamic acid. It also consists of cysteine amino acid. It also consists of glycine. These 3 amino acids combine with each other and form this amino acid complex. When Licotriene A4 is connected with this glutathione, it will give rise to Licotriene C4. And Licotriene C4 converted into Licotriene D4 by removal of glutamic acid. Once the glutamic acid that is attached with Licotriene C4 has been removed, we will obtain Licotriene D4. Licotriene D4 can further be converted into Licotriene E4 by removing glycine. I want to show you in picture form how this process is occurring in our human cells, especially in leukocytes. We say it leuco because leukotrienes are produced in our human leukocytes, but WBCs mostly. A4 means 4, it consists of 4 double bonds in the side chain. Here is arachidonic acid, consists of carboxyl group, 4 double bonds. Through 5 lipoxygenase, it is converted into 5-hepate as I have explained you. And there is a hydroperoxy group on the 5 carbon of this 5-hepate. This 5-hepate then further be converted into Licotriene A4 by conversion of this hydroperoxy group into this form, oxygen. This is the structure of Licotriene A4. This Licotriene A4 can be converted into Licotriene B4 by conversion of this into hydroxyl, and also conversion of some conversion also occurs at this carbon number 14, 13, 15. This is structure of B2. There is an addition of water. One hydrogen combined with hydroxyl, this oxygen to form hydroxyl, and one hydroxyl will be attached here. Licotriene A4 can be converted into by addition of glutathione into Licotriene C4. Licotriene C4 contain, this is a structure of glutathione, glutamic acid, glycine, and here is cysteine. As you know that amino acid cysteine consists of sulfur. Through sulfur it will bind to Licotriene A4 and results in Licotriene C4. When this glutamic acid will be removed from this Licotriene C4, this will give rise to Licotriene D4. And by the removal of glycine, this is the structure of glycine molecule that will be study the different type of amino acids in amino acid chapter. It will results in formation of Licotriene E4. So in this way, from arachidonic acid, we can get any type of Licotriene through lipoxygenase pathway. The difference between the lipoxin formation and Licotriene formation is on the basis of the enzyme. In lipoxin formation, there is a 15 lipoxygenase enzyme that results in formation of hydroperoxy on 15 carbon rather than carbon number 5. So as a result, 15 hepate will form. There will be some other enzyme 5 lipoxygenase, and the overall structure that results in lipoxin formation is that it consists of hydroxyl group on carbon number 5, 6 and 15 as well. So this is the simplest difference of lipoxins. But our major and the most important role that is performing by eicosanoids is because of licotrienes. Licotrienes are much more important as compared to lipoxins. Whereas lipoxins, it thinks that lipoxins can also act as anti-inflammatory substances, whereas licotrienes are the pro-inflammatory that involve in causing asthma and different type of diseases. Now we will discuss the functions of different type of prostaglandins, prostanoids. First of all, platelet aggregation that involve in formation of blood clots in our body, or if there is a hemorrhage of the blood vessels in the joint or different locations of the body, there will be a hemorrhage and platelets start aggregating there to form the blood clot. And how prostaglandins can affect the platelet aggregation or disaggregation. There are two types of prostaglandins. One are thromboxans and second are prostacyclins. Thromboxan A2 is produced by the platelets that are activated and involved in performing blood clotting process. So those platelets that involve in aggregation and blood clotting, they will also release thromboxan A2. What they will cause? This thromboxan A2 will cause different type of contraction or vasocontraction. As a result, they also cause the aggregation of further different type of platelets so that they can easily form a blood clot at the site of infection or at any other site. So thromboxans involve in blood clotting process by aggregation of platelets and by formation of blood clots. By vasoconstriction, they can also cause the vasoconstriction so that very blood will be lost at the site of infection. Whereas prostacyclin PGI2, prostacyclin is antagonistic to this. They cause the disintegration of platelets. If these are the platelets, they will cause disintegration of the different type of platelets. So they are against the functions of thromboxan A2. Second thing they perform, they cause the vasodilation, and they are produced by the endothelial cells of our blood. Our blood inner membrane consists of endothelial cells. These endothelial cells produce prostaglandin I2, which is called prostacyclin. They cause vasodilation, whereas thromboxans cause vasoconstriction. Thromboxan A2 causes the platelet aggregation, whereas prostacyclin, they are causes inhibition of platelet aggregation, or they causes platelet disaggregation. So both are antagonists to each other. Both functions are very according to their need, because thromboxan A2 are needed when there is a hemorrhage. But PgI2 is also necessary for the functioning of the body. If there is a clot, they prevent the body from having unneeded or wasteful production of clots in the blood cells. If there are a number of clots has been produced in the blood vessels, what will happen? They can cause different type of diseases, increase cholesterol level. Further, they can also disturb our heart and brain level. Thrombosis. If this thrombosis can also go to our brain and can cause blockage of the blood vessels or blood to brain, they can even cause a death. So prostacyclin are also very important. In that way, they causes the disintegration of platelets, and they not involve in, they dissolve the clot that formed by the thromboxans. They also prevent the unnecessary formation of clots in our blood vessels. I have also explained you how vasoconstriction is caused by different type of cells in the body. Like when there is a cell, this is a muscular cell, any cell, then calcium when incorporated or secreted by the if calcium enter from extracellular matrix as in our cardiac muscle, our sarcoplasmic reticulum also causes the release of calcium into this cell. This will bind with myofibrils and actin and myosin and results in contraction of the muscle. In this way, thromboxan also causes the increase in intracellular calcium, and in that, as a result, there will be contraction of smooth muscles and vasoconstriction. So this vasoconstriction process is also performed by other type of prostaglandins. Like prostaglandin F2 is abundant in uterus. Two other prostaglandins are also present or involved in performing functions of uterus. But prostaglandin F2 is most common. Like prostaglandin E2 is most common secreted by kidneys, involved in dilation of vessels in kidney or renal tubules. So as prostaglandin can also cause the vasoconstriction, so contraction is needed during the childbirth. These prostaglandins can also involve in contraction of or inducing the labor contraction in the uterus, childbirth or after menstruation, to remove the debris and the blood from the patient or from the mother or any female. So these to induce labor contractions. These prostaglandins also have a role in this, or to prevent the conception of the embryo. They can also involve in prevention of conception of the embryo. So these prostaglandins has different roles in uterus, stomach, kidney. The most important thing that I want to inform you, who Nicotrienes cause asthma. Our body consists of a number of mast cells almost in every part of the body, except in our central nervous system, brain, and spinal cord. During some allergic reactions, these mast cells are present in our lungs. When some pollen grain or some patient is suffering from engulf some any agent that can cause allergic reaction, these mast cells cause the secretion of leukotrienes. This leukotriene mast cell also release prostaglandins, other chemoattractive factors, but leukotrienes is also secreted by these mast cells. What is function of these leukotrienes? First of all, these leukotrienes can cause the vasoconstriction. If this is a bronchial vessel, they will cause this bronchial vessel to contract. This is normal diameter. This is though that diameter that is caused because of the action of leukotrienes. So leukotrienes causes the bronchoconstriction. As a result, there will be a difficulty in breathing in asthma patient. Second thing that these leukotrienes can perform, they cause the dilation of blood vessels. These are the blood vessels consist of different type of cells. There are capillary pores. They increases the pores. As a result, a large amount of fluid, plasma fluid, can got entered into this lungs. When a fluid is also enters in our lungs, what will happen? There is a large amount of fluid secreted by interstitial fluid secreted because of action of leukotrienes on the blood vessel. And leukotrienes causes the dilation of blood vessels, and a large fluid also accumulated, and further difficulty in breathing in the lungs, in expansion and relaxation of the lung. Second function. Third function performed by leukotrienes, that they also causes the attraction or activation of different types of eosinophil. Eosinophil tactic factor. They also attract other type of white blood cells like eosinophils. So three important functions performed by leukotrienes. As a result, leukotrienes will cause asthma. How we can prevent this asthma caused by leukotrienes? By preventing the enzymes that involve in formation of leukotrienes. We can overcome over this condition of leukotrienes temporarily. By inhibiting the 5-lipoxygenase, we can overcome over this disease. Or in normal cases, if it is caused by cause of prostaglandins, we use aspirin, ibuprofen, indomethacin. These are NSAIDs, non-steroidal anti-inflammatory drugs that can stop the formation of arachidonic or can inhibit the conversion of arachidonic acid into PGG2. There is no PGG2 or there will be no PGH2 if there is a block in peroxidase or cyclooxygenase, as aspirin do. How aspirin cause the inhibition? Aspirin consists of an acyl group. This is a structure of aspirin that consists of a benzene ring, carboxylic acid group, and through oxygen they bond with an acyl group. Aspirin causes the transformation of this acyl group into the cyclooxygenase. As cyclooxygenase is the enzyme, it is a protein, so it transfers this acyl group to the polypeptide chain through link with any amino acid. As a result, the global structure of cyclooxygenase enzyme will be disturbed. When the structure will be disturbed, then cyclooxygenase cannot perform or conversion of arachidonic acid into PGG2. There is no PGH2, there will be no PGE2 and other type of thromboxane etc. In this way, aspirin, ibuprofen and other types of non-steroidal anti-inflammatory drugs can inhibit the formation of different type of prostaglandins. And for inhibiting these NSAIDs cannot inhibit the leukotriene formation. For causing the leukotrienes to stop inflammation, we use inhibitors of lipoxygenase, not of cyclooxygenase. In our concepts, I have discussed the role of phospholipase A2 linoleic acid in eicosanoid formation or arachidonic acid. Eicosanoids cause inflammation. I explain you leukotriene, role of leukotriene and their role in uterus, kidney and lungs. Aspirin and cortisol. How can cortisol also involve in treatment of patient? Cortisol is a steroidal anti-inflammatory drug. It stops the function or it causes the inhibition of phospholipase A2. What is phospholipase A2? Phospholipase A2 that involved in conversion of arachidonic phosphatidyl inositol into arachidonic acid, that involved in release of phosphatidyl inositol, and that helpful in release of arachidonic acid that is present at carbon 2 of phosphatidyl inositol. So phospholipase A2 involved in formation of arachidonic acid. So when there is no phospholipase A2 functioning, there will be no phosphatidyl inositol. There will be no phosphatidyl inositol, so it will not be broken, and no arachidonic acid. No arachidonic acid, it means that there will be no any type of further arachidonic acid. So cortisol decrease the formation of arachidonic acid. Low arachidonic acid, low prostaglandin. This is a simple thing. Aspirin, indomethacin, ibuprofen causes the blockage of conversion of arachidonic acid into other forms, whereas the cortisol involved in inhibition of formation of arachidonic acid. So one medicine involved in inhibition of arachidonic acid formation, and second involved in inhibition of conversion of arachidonic acid. And I have also explained you how thromboxanes and prostacyclins work. Thromboxanes and prostacyclins are antagonistic to each other. One causes clotting formation and other inhibits the clot formation in our body. For reducing inflammation or treating any type of disease, if a person is taking aspirin and other types of anti-inflammatory drugs for a long time, what will happen? There will be no production of prostaglandins. No prostaglandins. If there is a very low production of prostaglandin for a long period of time, the kidney functioning, the uterus functioning, and stomach functioning, these will be affected, because normal secretion of prostaglandin is necessary for the working of uterus, for contraction of stomach, for renal tube functioning of the kidney. So this is also a side effect of long-term use of aspirin on these organs of the body. This is the today's lecture of chapter Lipids, lecture 3. Hope you have understand what are the eicosanoids, how they are produced and their role, and what is the role of aspirin, how aspirin can reduce inflammation, ibuprofen, etc. And different types of prostanoids, leukotriene synthesis, leukotriene types, how they form. Thanks. Give us a like, subscribe. Thank you. Allah Hafiz. Share as well.